So good morning, everyone. It's now 7 o'clock here in Nashville and many times at many places. So good morning, good evening, and everything in between. We're going to wait just a minute or two to give everyone a chance to log in. So just hold your horses and we'll get started here in just a couple minutes. Lila, where are you at? Are you in Florida or in Massachusetts?
Massachusetts. We stopped going to Florida when the pandemic started. Actually sold our house. So now we put up with winter, but we actually like winter, so it's okay.
Oh, good. Yep. It's probably high autumn there right now, right?
It is. It's just at the later part for the foliage right now, but it's beautiful weather. We had spectacularly, it's freezing in the morning and then it gets to 60 or 70 during the day. Yep. Perfect. My favorite weather.
Yep. Okay, well, I think maybe we'll go ahead and get started here. So again, good morning, good evening, and good everything in between to everyone. And welcome to our Protein Society webinar for this month, which is exploring proteins in living cells. And we have a distinguished and international panel of speakers, and we're looking forward to hearing from them. I'm Chuck Sanders. I'm the president of the Protein Society. And I just want to offer a couple of introductory words about the society and before we move on. And so I want to remind everyone that the Protein Society sponsors a journal, Protein Science. We have a relatively new editorial team led by John Curian, and we hope that you'll submit your best work to this journal. One of our associate editors, senior editors, Aitza Lopez Cora Harina, is here, and she's going to say a few words in just a moment. So I'll turn it over to her. First, let me say, though, I hope that you'll consider attending our annual symposium this summer in Boston. We usually have it there every other year, and it's July 13th through 16th this year. And these are really wonderful symposia. And if you can fit it into your schedule, we'd love to have you. And so thanks very much for attending this event today. And I'm going to turn it over to Aitza now to say a few words about the journal.
Okay, so good morning, evening, and depending on your time zone. I would like to very happy to be here, and as a kind of Chuck mentioned, as a senior editor of Protein Science. So just I would like to introduce you a bit the journal. So Protein Science is the Protein Society journal. It's a widely journal. And here are the web pages where you can find us. A bit the scope of this journal is obviously focused on protein science. So, and it's an international forum, and this I like to highlight how international is the journal and the team for publishing these original reports on scientific aspects of protein molecules. We would like to have this as a kind of a community journal for all the scientists working on protein science, and we'll try to be also very open to not only structural biology work, but all the work related to protein science. A bit like the journal is kind of a journal, but I think one of the most important things of the journal is the team that conforms the journal, and we'll be looking to the papers that they are submitted. So as Chuck mentioned, so we have John Curian as our editor-in-chief, and we are two senior editors, Nir Bental from Tel Aviv University in Israel, and myself, that I'm from Zikbi Magune Research Institute in Spain. We also have several associate editors, and And they are also kind of rather international. So we have Professor Akutsu from Osaka University in Japan, and Zeng-Yi Chang, that is also, I would like to thank him for organizing this session with this particular focus on Asian scientists. So, and we also have Janine Amerter and Carol Post. So I would like to highlight a bit this wide expertise that all the protein science team compiles and the diversity of the team. We also have an editorial advisory board, and I will not go into the details, but we have this extensive list of highly recognized scientists that they are part of the board, also helping with the journal. And we have a new kind of concept that we are very happy to present, that is this early career review board. So it's coordinated by Janine Amerter. And here what we want is to involve scientists at the very early career stage in the peer review process and also in the scientific publishing processes. So we put together a team of excellent early career researchers that they are kind of helping us with this board. I would like to show you a bit kind of the diversity of topics that we are publishing, and kind of one of the examples is those special issues. So we have a yearly tools special issue, very kind of high impact kind of journal, and what we are compiling there is like those publications like focus on tools related to protein science, from computational to experimental new tools and approaches, and this is kind of coordinated by Nir Vental. And we have different currently open special issues. So if any of your research fits into these topics, please consider submitting your manuscripts to the journal. In addition to all these special issues, so obviously we are open to new contributions and exciting scientific manuscripts. And a bit, we want to be kind of a close kind of community journal, and we want to make the life of our authors easy. So therefore, we have implemented this free format submission. So you don't need to put a particular format to submit to our journal. And we also implemented this continuous online publication of articles. As soon as they are accepted, they are kind of published online. We also want to remind you that the Protein Society members, they have these reduced publication fees for publishing in our journal. And with that, I would like just to thank you. And we are looking forward really for your submissions. Thank you.
Okay, so I think we're going to turn it over now to one of our Wiley partners, April, who will introduce our speakers, and we'll get the scientific webinar and ruling here. April.
Hi, everyone, and thank you for the great introduction to the journal, Eitzeber. Welcome again to the Protein Science Journal's webinar on exploring proteins in living cells. My name is April Rod, and I'm a development editor at the journal, working at Wiley, the publisher. I'm very excited that I had the opportunity to work with Dr. Zeng-Yi Chang, an associate editor on the journal, to organize this webinar today, specifically for a global audience with an international speaker list. This webinar has been the result of a collaboration between the Protein Society, who is also our host, the Chinese Protein Society, and Wiley, with me at the publisher side trying to coordinate things. As a reminder to all of our participants, use the Q&A to ask questions, and after each talk, we'll be able to answer a few of them, but the speakers will also try to answer them in the chat as best they're able as we go through. We have 3 very excellent research talks today, and our first talk is from Dr. Lila Gierasch from University of Massachusetts Amherst in the US. And her talk is how selectively promiscuous binding enables HSP70 chaperones to facilitate folding in the cell. Lila, do you want to get started?
Okay, let me get the right screen shared. All right, everybody can see that?
Yes.
So good morning, good evening, good whatever. I really thank the Protein Society for starting these webinars. I think there have been some very beneficial things that counterbalance some of the horrible stuff we've been through with the pandemic. And one of those is the ability to connect remotely. And it has facilitated a lot of our science. And I think this is a wonderful example that we've realized we can continue it and link ourselves across the globe. So it's great. It's a pleasure to have the opportunity to tell you a short story about some of the science going on in our lab and how it illustrates some of the challenges that are presented when we do protein science in the cell. So I'm going to get rolling. And I want to first start by saying that there are several heroes behind the work that I'll talk about today. I want to highlight, let me get the laser pointer going. I want to highlight the two folks at the top, Sasha Pozidaeva and Eugenia Clerico, who really were spearheading the work. It was started initially by a grad student, Joe Talitsky, and undergrad Rachel Jansen played a critical role, and John Osden from Meg Stratton's lab at our institution helped us in a key way that I can mention as I go along. So what's the problem? The problem, I think, that we have to face when we look at proteins in the cell is that it's a living organism that we're talking about, and there are many challenges that are presented to the proteome of an organism, and they're unavoidable. What this means is that we need rescue operations to be going on at all times. And I just made a little list of the things that you have to think about when you're thinking about the challenges to proteins in the cell. Biosynthesis is imperfect. We know that there are mistakes made. Stress is unavoidable. Mutations happen. This is a key part of evolution. And so there are mutations that challenge the stability of proteins. Proteins are dynamic. They are not meant to be rocks. And so functional requirements ask them to be able to sample many conformations. This leads to metastability. Otherwise, generally functions would be compromised. Some functions require full unfolding, for example, translocation across membranes. And then we have the inevitable accumulation of damage to proteins that happens with age and time. And this also is accompanied by a diminution in the robustness of some of the capacity of a cell to deal with problems in the health of the proteome. So it should be no surprise that we actually have going on in our cells at any given time a huge array of processes whose role is to support the health of the proteome and to make sure that if their proteins become so damaged that they can't be rescued, that they get broken down. All of this makes up what we call proteostasis. We look now at the folding problem in the cell in a different way from the way we used to. We have the wonderful idea that there's an energy landscape that describes how proteins folds, and that in navigating this landscape, proteins can sample intermediates. But we've also recognized that some of these intermediates have stability that leads to a longer lifetime, and that any of these states that are not fully folded are susceptible to intermolecular interactions that compete with folding to the native state. Of course, I'm talking about soluble proteins at this point, although many of the things I say would apply also to membrane proteins. Competing with folding then will be the processes that happen when two proteins get together and expose interactive surfaces, and that's aggregation. And aggregation can lead to, we know, organized fibrils, oligomers, or amorphous aggregates, but all of these are really hyperstable because they have many strong interactions. So what we have going on at all times in the cell is an effort to diminish the likelihood of these processes. We have chaperone networks, and I'll say more about it, but these chaperone networks basically facilitate the exit from partially folded states that are highly or quite stable, so they're populated. Also, chaperones can inhibit the processes of intermolecular interactions. And so we look at this as a constant process of rescue to help folding happen with greater fidelity. One of the chaperones that is at the hub of the networks of chaperones that are present in your cells are the HSP70s, and the rest of what I'll talk about relates to this family. But what I'll say applies to many chaperones. HSP70s are workhorses. They can actually facilitate many processes in the cell, which is one of the really stunning questions that they present. So HSP70s greet nascent chains. They do this process of maintaining a protein in an unfolded state so that it can go across a membrane. They can help rescue misfolded proteins. They can help a process of disassembly from aggregates. And they can actually, in some ways, facilitate folding. All of this is done by the same machine. In addition, one of the things that I want to get across is that we have the chaperone networks that handle the entire proteome. And so it's a network of species like the 70s that have to interact with many, many, many different substrates. We often call these clients. And this underlies the questions that I'll be telling you about today. How is it that a network of chaperones can work with the whole array of proteins in the proteome? And for the 70s, I'll talk a little bit more about how the machine works and then focus on how they interact with their substrates. So the machine itself is a wonder of allostery. HSP70s are two-domain proteins. I'll show you the structure in a moment. And they have a nucleotide-mediated conformational change. In the presence of ATP, they visit a low affinity for substrate state. And in that state, the two domains, the nucleotide binding domain, which is an actin fold, and the substrate binding domain, which I show in green and red over here, are intimately docked on one another. In the binding of substrate and the interaction with a co-chaperone from the J protein family, the ATPase is activated and one gets nucleotide hydrolysis and the switch to a conformation that has very high affinity for the substrate. It's locked down between this green domain and a helical lid, and I'll show you again in the structure in a moment. The high affinity state then switches back to the low affinity state through exchange of nucleotide facilitated by a nucleotide exchange factor so that ADP is exchanged for ATP, the substrate is released. And this cycle is really, as I say, a wonder of allostery because the binding of the nucleotide is what causes this large conformational change and the cycling between a low and a high affinity state for substrate binding. It is this ability to cycle between low and high affinity that allows HSP70s to perform all of the functions that I just showed. And that's really an amazing thing. But if you think about it, binding of segments of a polypeptide chain and releasing them can delay folding because that polypeptide cannot get to the native state, can also help disassembly or disaggregation. So it's a machine that nature has come up with to facilitate the processes that I talked about. Specialization generally is governed by the J protein and a bit by the nucleotide exchange factor, but the heart of the machine stays the same for all of the functions I described. I promised you a look at the structures. So the low affinity state is on the right. We are now fortunate to have a crystal structure that describes this state, although it took a long time coming because it's very dynamic and had to be pinned down by a crosslink to crystallize. So the nucleotide binding domain, as I said, is an actin fold. This is the intimate docking of the substrate binding domain with its helical lid lifted off and binding to the nucleotide binding domain. When substrate binds and ATP is hydrolyzed, the two domains dissociate from one another. There's a flexible linker and they behave as rather independent domains. The substrate binds, as I said, in this green beta cradle with the helix lying over it. So in going to the low affinity state, we have the helix lifting off, the substrate binding domain now interacting intimately with the nucleotide binding domain. It's a fabulous conformational change. And in terms of allostery, it's amazing that all of this happens because a gamma phosphate is present on the ATP. And I'm showing you what a mammoth conformational change it is here and a morph between those two structures. So this is the machine. And we have some understanding now that's pretty darn good about its allostery and how it cycles back and forth between high and low affinity. But one of the things that's a major puzzle still outstanding is how it is that the binding to the clientele of the HSP70 enables it to carry out its functions. And we like to call the binding, because it has to occur to many, many different proteins. It's a selectively promiscuous binding, and I'll say that word again several times. Because basically what we know at this point, largely coming from binding studies with peptides, is that many but not all sequences can bind. So that's the selective promiscuity. Preferred binding sites are hydrophobic, and there is a preference for positively charged residues. In fact, a bias against negatively charged residues. The binding sites, when you look at, and there are predictive algorithms based on array studies, when you look at them, they're predicted to occur every 40 residues in the proteome. So this is not like a limited capacity to bind. On the other hand, what's special about these binding sites is that they would be sequestered if a protein is folded in its native state, because you're looking for hydrophobicity, and this bias towards positively charged residues gives a little further selectivity. In addition, if you were to do aggregation predictions, you'd find that the binding sites for HSP70 coincide largely with aggregation-prone regions, which makes teleological sense. So they have hydrophobicity and often a high beta propensity. But when you think about this, it is a selectivity because they would not interact with native proteins, but it's a promiscuity because these sites are not highly restrictive. So that's the kind of term of the day that I want you to think about. So the question is, how does that binding take place? If we look structurally do a little deeper dive, this is the substrate binding domain that I was showing you with the green beta-rich domain and the helical lid, and the substrate binds in a little binding site created within the beta subdomain. If you take off the helix and look from the top, you get the picture on the right. And what we can see, and this came first from a crystal structure back in 1996 from Wayne Hendrickson's lab, is that the substrate is in an extended conformation, and really 5 residues have intimate interaction with the chaperone, which is quite a small number of residues to create this binding capacity. There are identifiable binding sites for the side chains and hydrogen bonding with the backbone that leads to 5 principal hydrogen bonds. The red pocket here in the center, which we call the 0th site, is the one that looks most lock and key, and it prefers strongly a leucine, although isoleucine, valine, or proline could be accommodated. The other pockets are really not even pockets, they're like shallow pockets, and they have some degree of preference for binding to particular side chains, but it's not super strong, and I'll say more as I go along. The -1 and -2 sites from peptide array studies seem to be more stringent, and I'll say more about that as I go. I'm going to call this arrangement of the substrate in the binding site now N to C terminal. And of course, it depends how you look at the substrate binding domain, but just stick with that as what we'll call the canonical binding orientation, which was seen in the first crystal structure and in many, many other structures. So what we wanted to do was to move from that picture that came from binding of a peptide and many, many of the other studies that are done on peptides to see what happens now if we do a study of a precursor, a unfolded protein that the chaperone can bind to that will have multiple binding sites on it. And are the things we learned from that crystal structure with a short peptide and the other structures that have been done applicable when we look at binding to the whole client? So we picked a protein to do this on that has been studied in Babis Kalodimos' lab. It's the precursor to the periplasmic protein alkaline phosphatase in E. coli. And we're working with the E. coli HSP70, which is called DNA-K. So I'll use those names interchangeably. Why pick Pro-Fo-A? Well, what Babis showed us in his work is that this protein is soluble in the unfolded state, which biophysically makes it a good subject for study. It's stably unfolded if you reduce its 4 native disulfides, and we went further and actually mutated the 4 native cysteine, the native cysteine residues to serine. So we have Pro-Fo-A with serine substituted for the cysteines. And it's shown to interact with DNA-K in vivo, which means that it's a substrate that is not lacking in relevance. It's not clear how critical this is in the secretion pathway of the protein, but it has been shown in Carol Gross's lab several years ago that there is an interaction in the cell. So what we wanted to do first in the study that I'll tell you about was to validate that this is a substrate that one can use as a biophysical substrate, and DNAK binds to it in vitro. And then we wanted to use peptides as tools to examine the potential binding sites in this protein with greater detail. And we've done several biophysical methods, X-ray crystallography, we've used NMR, and we've used some clever biochemical experiments created by Eugenia and Sasha, because we wanted to make sure we understood the orientation of the substrate when bound. And then what we do is cycle back to the protein level to see if what we've learned from peptides is giving us a picture of how the chaperone binds to the protein when it's presented to the full length of the protein. So one of the major tools that's been really helpful in our work is methyl NMR, and in particular, isoleucine delta methyls, because it turns out nature has given us the gift of two isoleucines that are right adjacent to the bound substrate. For reasons that we are delving into, but at the moment don't understand, is that these two isoleucines present reporters in these delta methyls with very strong chemical shift differences depending on the nature of the peptide that's bound. This allowed us to do a titration where we present to the substrate binding domain of DNAK increasing amounts of Pro-Pho-A and ask whether we can see binding in vitro. The binding is accompanied by a shift from the black signal, which is the free substrate binding domain, to the bound forms in blue that are clearly experiencing chemical shift differences. Sorry about the phone in the background. That are quite striking. This is the two isoleucines that I show in the diagram up above, 438 and 401. For the moment, what I want you to take from this is that it's an indication of binding. Furthermore, it's an indication of binding that shows as you add more substrate binding domain, you get new signals. So it looks as though there are multiple binding sites. So now we're going to go ahead and say, can we use peptides to understand what this binding interaction is in greater detail? To come up with the peptide models, we looked at array data that had been published several years ago from Stefan Rudiger's lab that pointed to sites. Again, this is an array, so these are presented as peptides within the sequence of Pro-Pho-A. There are 13 mers overlapping by 10. And so basically this array data pointed to a number of sites that are potential binding sites for DNAK on the Pro-Pho-A substrate. I highlight in red how we took the intensity data and identified what we thought were likely sites. We also used predictive algorithms. And then what we did was to go ahead and make these as peptides. The length of the peptides differs primarily so that we could come up with a soluble peptide that was a good model to use. And we could also go ahead and take these peptides and see, did they bind and what was their affinity? So the affinities is shown over here. The strongest binding actually corresponds to the signal sequence, which we might want to talk more about later, but it's clearly a very hydrophobic sequence. And there really are hydrophobic sites within these peptides, not surprisingly. These sequences are quite dispersed within the Pro-Pho-A overall structure of 400 and some odd residues. This is a point that I'll return to at the end, and if I forget, please ask me a question about it. But we could then go ahead and ask about how these peptides are binding and get structural detail at the level of the individual residues. And this really hasn't been done in the past. So first off, Rachel Jensen, the undergrad in the lab with John Osden, were able to do crystallization. And I'm only going to show you a couple of examples, but we actually had 10 crystal structures of these different peptides in different lengths and forms bound to the substrate binding domain of DNAK. They do bind in an extended conformation, as was seen in the original Hendrickson structure. But the first surprise, these were labeled, you might have seen A, B, C, D, E, is that if you look at peptide C in the crystal and peptide D in the crystal, they actually are in the opposite orientation of the polypeptide. And it was known that this was possible, but it was felt to be the unusual case And in the past, generally only when proline was enriched in the sequence. But here we are with two good garden variety sequences from Pro4A, and one of them is selected to bind in the N to C canonical orientation and the other in the C to N. How is this possible energetically? Well, I want to point out down below on this slide that in the substrate binding domain, the binding is actually occurring where there's an unrequited beta strand. So in one case, what we call the canonical, there's actually a parallel beta-beta strand-strand interaction with the normal hydrogen bonding taking place. And in the other case, it's anti-parallel. And as you learn in basic biochemistry, both of these arrangements can form backbone hydrogen bonds that are reasonably good. And so we get the 5 typical hydrogen bonds in both arrangements. In addition, even though the side chains point in slightly different orientations, they each can sample the pockets. And the leucine in the central pocket is present in both of these structures. And this is what we know is like the anchor residue. So this was the first kind of surprise, and it's from crystal structures. So we've then had to move on and say, is this happening in solution, or was it by chance something that was stabilized in the crystal? So Eugenia and Sasha came up with 2 experiments to show orientation in solution. One is a paramagnetic relaxation enhancement experiment with a cysteine on the end of the peptide labeled with a spin label. One could find whether which side of the substrate binding domain was broadened when the peptide bound. It's shaped kind of rectangular with a thickness to it. So it actually lends itself to seeing the sidedness. The other experiment involved forming a disulfide crosslink, putting cysteines on the substrate binding domain on either side and finding out for a cysteine in the peptide, which side it could form a crosslink with. So these 2 experiments were really very elegant ways of showing orientation. So then we move into the solution regime for these 2 peptides, where we have in solution the question, do they bind in opposite orientations? First thing is NMR shows us through these isoleucine chemical shifts that we have one bound form in solution, and for each of the peptides, which is already a fascinating thing. Particularly peptide C has 3 leucines, but one form is favored. And both of these forms now we can follow by paramagnetic relaxation, and we see that there is broadening in the case of peptide C on one side and the opposite side for peptide D. And so this, together with cross-linking that showed cross-linking on one side for D and on the other side for C, this tells us that the solution conformations reflect the same arrangements as in the crystal, that one of these is forward and one of these is reverse. So this was really cool, and we were very excited. And it opens up a different kind of view of the way the chaperone can choose to bind its substrates. It's not the only story, though. If you look at another example, peptide E in the crystal bound in the canonical forward direction, it's extended, puts an isoleucine in the pocket, but otherwise pocket occupancy looks like what we'll call a typical canonical binding. We put it in solution and we find there are 2 bound conformations. And so the chaperone says, these are nearly isoenergetic. One is a little preferred, and one of them is forward and one of them is reverse. So this shows us there could be heterogeneity in the way of binding, in addition to orientation being a variable that we hadn't appreciated before. So I'm not going to give you more examples, but we went through all the peptides that are present in Pro4A as potential binding sites. Oh, sorry, I wanted to show you can get both cysteines cross-linked. When we went through all the peptides, I said to the panelists earlier, I want to put my favorite slide back in. This is my favorite slide because we learned by looking at all these peptides that they give diagnostic chemical shifts in these isoleucines for the residue that's present in the zeroth pocket and the orientation. And because we had a whole array with supporting data from crystallography and the biochemical experiment, we actually have fingerprints so that we can look at a new peptide and get an idea how it binds. And this has been a workhorse for us. It's really quite stunning. When we then look at all the peptides, we have these signatures for how they bind. We want to move back to the protein. So here's the titration that I showed you earlier, adding substrate binding domain to the full-length Pro4A. And we overlay the new signals that we see for the substrate binding domain on the signals we saw for the peptides. And what you find is initially you get new signals and they overlap with the site A, which happens to be, from that affinity table I showed you, the highest affinity binder of peptides. So this is like an aha moment. It looks like the chaperone scans the sequence of the protein, finds its favorite site, and occupies that first. We did have a surprise, which we haven't answered yet, that there's a novel site, which we call U because it's unknown at the moment. We're working on it. Then as you add more substrate binding domain, you see a new site coming that overlaps with C. And as you keep adding, you get sites that overlap with E and D and B. And so what we find, there's several messages coming from this titration. First, the overlap when the chaperone binds to the protein substrate with the peptide data is really strong. So it's binding as though it's seeing an additive sample of all these peptides when it looks at the unfolded protein. Secondly, there's a hierarchy of binding that is really based on the affinities for the particular sites. And by the way, this includes the orientation that is preferred in our studies on the peptides appears to hold when the protein binds, which is a really important finding. And so we have work to do on the novel binding site, but otherwise we have some really strong lessons that come from this. If you look at all the data that are embedded in that titration, these are the binding arrangements that we see in the protein, and they mimic what we saw in the peptide. You can see that in several cases, A and B and E, we have multiple binding modes that are possible. We couldn't tell between these two. If you look at the central residue, it's usually leucine or isoleucine. We have one example that appears to be proline. We can look at sites -1 and -2 and even out to -3. You have more similarity in the character. They're hydrophobic or positive, whereas sites +1, +2, +3 are less stringent and accommodate a wider array of residues. This was indicated by arrays, but now confirmed by this deeper dive. And so this is the picture we have. What does it say to you? I want you to look at the bottom of this slide first. What it says to you is that we have a fluctuating, very dynamic situation when a chaperone, this chaperone binds to an unfolded protein. It can sample any of a number of sites. It appears to do so based on their affinity. It can scan and bind in either N to C or C to N orientation. And we were looking primarily at substrate binding domain, which is high affinity, but in the presence of the full-length chaperone with nucleotide present, these binding sites, as I showed you in the allosteric cycle, will be bind and release processes. And so the maintenance of an unfolded protein would be through binding and release to multiple sites, and these would be in a fluctuating dynamic nature. If you think about this energetically, these are tens of micromolar to one case was nanomolar. They're still relatively weak binding. The differences between the energies of binding for different sequences, we seem to see that it's small. In many cases, there are multiple binding modes. So there's small energy differences between different binding modes, dynamic sampling of multiple binding sites, and when we have the full-length chaperone, nucleotide modulation of how long the chaperone will stay bound. We would like to then move you back out to function. So how does this help function? We think the hierarchical binding would be co-evolving with the nature of the sequence of any client. And this could lead to the release of some binding sites before others, and a coupling of the optimization of the functional roles of the HSP70s with these biophysical characteristics of the binding. If you think about what processes I mentioned could be facilitated by HSP70s, folding itself, the mechanism of folding, and in fact, there's a recent study from Ulrich Hartel's lab that shows that in the folding of luciferase, DNA-K can facilitate both the kinetics and the yield. And he posits that it is by binding a particular site that is prone to aggregation. This again would be an example. If folding is optimized, it could be because the hierarchy of binding favors sites that can make the folding reaction occur with higher fidelity. Many events in the cell involve multiple chaperones, and the handoff from HSP70 to a downstream chaperone could be facilitated by the nature of the binding site and the order of release. This would be the same for degradation machines. Frequently, HSP70s are at decision points between giving a protein a new chance to fold or go on to a next chaperone or to be degraded. And then in secretion, you can imagine the partnership with secretory pathway components, or in disassembly, could be facilitated by this hierarchical orientation independent binding. So we really think this is making us think differently about how the chaperone works. There's a lovely example from David Agard's lab of handing off to a HSP90. And in fact, the sequence that's bound is bound in what we call the reverse orientation. And this allows the glucocorticoid receptor ligand binding domain to have a productive interaction with the next chaperone. So with that, I want to end by thanking the folks in the lab in a more group-wise way. It's a great group. I mentioned the people that were involved in the work I talked about today: Eugenia, Sasha, Rachel, and John, with the beginning starting with Joe Tulitsky, but the ongoing work is being done by those in red, alums of the lab in blue, and then our collaborators. And we did get some wonderful help from John Han Chen and Eric Nordquist to do a new prediction algorithm based on our findings. So with that, I am happy to expand on any of this, answer any questions, and turn it over to all of you.
Thank you, Lila, for a great talk. We have a question from the Q&A. Any comment on the on-off rates for HSP70 binding and their implication in the chaperone function?
Yeah, it's a great question. I can tell you that the off rate in the absence of co-chaperones is quite slow. One has to rip that helix off, and it has salt bridges and hydrogen bonds. In the presence of co-chaperones, it goes from tens of minutes to usually the turnover time is on the order of tens of seconds. So that is a more typical kind of value, but it's going to vary depending on the nucleotide ratio between ATP and ADP and the affinity of the particular site, because the binding interaction for the substrate stabilizes the high affinity state and so keeps the chaperone in that state longer. We're in the process of modeling this. Others have modeled it, but those are some general comments. And clearly, when you have the subsequent chaperone in a handoff or other processes taking place, they will also contribute to the release. So it's one of these things that'll be tuned in physiology. It's not, we can't look at some hard and fast numbers, which we think is actually part of the fascination.
Great, and we have time for one more. This one starts, congratulations to you and Eugenia on wonderful work. Is it possible that HSP70 would bind to the target polypeptide and be processive? And is binding in a slow exchange regime?
Yeah, so clearly those who do NMR know that in the experiments I showed, we're in the slow exchange regime because we see the bound state and the free state separately. That is a very artificial system because it's the substrate binding domain in the substrate model. And so I really think we would move in, if we have the whole system cranking, we would move into intermediate exchange. But we haven't actually looked at that by NMR because it's a pretty tough system to look at. We do hope to. It would be a lot of fun. Is it processive? We have no experiments that indicate that, and it's my kind of belief at this point, because I can't tell you an experiment one way or the other that is not processive, because the binding and release really looks as though what we have is kind of a stochastic search by encounter for the highest binding sites, and then a release, so that it's because of that dynamic transient nature of binding, there will really be, it's not an equilibrium because we have nucleotide hydrolysis taking place, which makes it a more vectorial process. However, we do have the chaperone in its low affinity state able to encounter many different sequences based on diffusion. So there could be some systems in biology where there's a directed binding, but I don't see this chaperone as being an example of a processive case.
Thank you,
There is
Lila.
another question there. I don't know if we have time, April, about the signal peptide.
I think we have to move on, but as we
Okay.
move forward into the next talk, if you could tag your questions for Lila, if they're for her, so this way we can differentiate between that and our future speakers. I think that will make it much easier for you to answer questions in the Q&A section. So up next, we have Dr. Yutaka Ito from Tokyo Metropolitan University of Japan, who is staying late for us to give us this talk.
My pleasure.
And his talk is on solution NMR approaches to 3D structure determination of proteins in living cells. And take it away. Thank you.
Okay, thank you. I'm going to share. Okay. Okay, so my presentation appears properly.
Yes.
Okay. Yeah. Thank you very much for the kind introduction. I'll also thank the Zeng-Yi and the Protein Society for providing me this opportunity to speak here. Okay. And I would start my talk with an episode for approximately 40 years ago. I was a PhD student and working on NMR studies of human HRAS. After determining the solution structure, NMR structure of GDP bound state, we started tackling of the active state, GDP bound state of RAS. And it turns out the NMR signals due to residues that are located around functionally important regions were not observed. That these regions are effector regions, which are responsible for target recognition on the alpha beta phosphate binding site or the gamma phosphate binding site or GTPase activity center. So the further NMR experiments revealed that there are multiple conformations exist for that region, and which is, the signals are extremely broad because of the exchange contributions. So for the flexible regions, that we have a conformation of multiplicity, and exchange rate was estimated to microsecond to millisecond time scale, and for the biologically important loop regions of HRAS-GTP in solution. So the conformation, we thought that conformation of these flexible regions may be easily altered by various interactions. And we had a question, what is a real conformation or conformation equilibrium of the flexible regions under extreme molecular crowding environment in cytosol? Of course, there was no terminology called molecular crowding at that time, but we have some kind of a doubt about the real image of these regions in cell. But unfortunately, there was no tools to investigate proteins at work in a living environment at atomic resolution, but now we can do it by incident NMR spectroscopy. Okay, so in this figure, I'm just show how proteins feel the intramolecular environment. First, I would introduce Dr. Sugita in RICAM reviewed that the MD simulation that changes in structure and dynamics of water surrounding proteins. And of course, there are excluded volume effects, very well-known one, and which let proteins to take the most compact native structure. On the other hand, there are non-specific interactions which tends to make proteins to have relatively extended structures with wider surface area. So, so this excluded volume effect and non-specific interactions are somehow pulling your proteins in an opposite direction to stabilize or unstabilize or your protein. And of course, there are specific interactions. So we can say that protein structures and dynamics in cells exist under a fine balance of these effects. And if we are very interested in fine details of protein structure and dynamics in cells, apparently NMR is one of the ideal methods because NMR has non-invasive character and ability to provide data at atomic resolution. Okay, so Okay, so for ordinary solution NMR approaches, we do stable isotope labeling with carbon-13 or nitrogen-15 and purify the sample for NMR use. But for incident NMR experiment, we actually use the cell suspension, live cell suspension for NMR experiment. So if we successfully label the target proteins with stable isotope carbon-13, N15 or F19, whatever, NMR active nuclei selectively, even under intracellular environment, you can measure heteronuclear multidimensional NMR spectra selectively, and which provides a good tool for monitoring structure changes, post-translation modification, substrate binding, or protein-protein interactions. And this figure is actually adopted from the, I forgot to say that incident NMR is first proposed by Volker Dutch when he was in UCSF. And the first publication was actually appeared in 2001, the beginning of this century. So actually this figure is adopted from his quite early review paper. And in the review paper, these 4 applications were actually described as a kind of a challenge in the future perspectives. But nowadays, after 20 years since the beginning of incident NMR, these events has already been demonstrated with the various proteins by incident NMR. Okay, so among these applications, our group has been interested in the structure changes. Okay, so at this point, I would show you how we could prepare samples for incident NMR experiments. So Volker used the protein expression systems in E. coli. Then toward extension of this methodology to eukaryotic cells, the various methods has been proposed. For example, microinjection to genopus oocyte, or cell-penetrating peptides, a pore-forming toxin electroporation for cultured mammalian cells. And very recently, the new method by using the microfluidics has been introduced. And of course, the protein expression systems in cultured mammalian cells or insect cells are also useful for incident NMR works. Okay, so as I mentioned to you, we have been working in incident NMR world approximately more than 20 years. So the accumulation of knowledge about protein behaviors can be categorized into, from my point of view, roughly these 3 points, just for the protein behaviors. The first one is destabilization of protein folding. So in the year 2009, Masa Shirakawa and his collaborators showed a very nice, very interesting paper describing ubiquitin fold are destabilized in HeLa cells. So they used they introduced ubiquitin to HeLa cells and looked at a so-called HD exchange. But I would just make the story very simple, and just would like to remind you that where we have enhanced amide deuteron to amide proton exchange rate means the hydrogen bonds or the secondary structure around the region are somehow destabilized. Okay, so to make the story very simple. So this is the data. So these points represent in vitro case, very slow exchange rate. But in contrast, you can find in cell, the exchange rates are 15 to 22 times faster. So it suggests a destabilization of protein folding. And this tendency will appear in all over the protein. So it was concluded that destabilization has happened on all over the ubiquitin fold. And also they used the A mutant, triple mutant, so as to reduce the affinity to ubiquitin binding proteins drastically. And eventually they concluded that the both specific, well, in this case, specific binding to ubiquitin recognizing proteins and non-specific interactions somehow contribute to the destabilization of ubiquitin. So it's these findings are quite sensational, and because we have thought that the macromolecular crowding inside cells somehow stabilize the protein folding by the excluded volume effect. So this finding is quite new. And then I'm going to overview the related topics by other groups. So, and then the in vitro NMR experiments appeared from the Gailey pilot group, and they utilized this protein, and again, HD exchange experiments are performed. And I will just make the description very short. They found when they used synthetic polymers like polyvinyl pyrrolidones as crowders, they found that protein folding stabilization. And in contrast, protein crowded BSA or lysozyme had a destabilizing effect. So it's very interesting. And they actually used this cytosol itself for this kind of experiment. And they found that the cytosol, E. coli cytosol, caused the concentration-dependent destabilization of this protein. And the effect of the cytosol are even stronger than those of BSA lysozyme. And so this is very good point that the intracellular environments are really significant, has a significant effect for the destabilization of protein. So then our thermodynamics experiment by utilizing incident NMR appeared from a Swedish group, Jens Daniels and others. And they, in this paper, they used a superoxide dismutase 1. And so SOD1 has a stable dimer conformation, but when you modify the loop lesion, you can have a SOD1 barrier, which has a stable monomeric species. And if you look at the NMR data of this SOD1 barrier, which can show you a single species corresponding to the completely folded species. But if you introduce one additional mutation, an I35A mutation, suddenly we see two pairs of cross peaks, well, one residue corresponding to the denatured state and the folded state. So approximately 1-to-1 equilibrium. And what is more interesting is when you introduce this mutant protein into a intracellular environment in the eukaryotic cells or in E. coli cells, suddenly equilibrium shifted to the unfolded state. So somehow the intracellular environment actually stabilized the unfolded state or destabilized the folded state. I'm not so sure, but still, and the intracellular environment has definitely had that kind of tendency, and which is observed in the NMR spectroscopy. And finally, in this point, I would show you an MD simulation, the all-atom simulation from the Yuji Sugita's group and the collaboration with Michael Fagan, Michigan State University. This is a all-atom simulation of the bacterial cytoplasm. So they created all possible protein structures by homology modeling and made this kind of system. And one interesting thing is when they looked at the A protein called a pyruvate dehydrogenase, which is representing green in the center. So during the simulation, the folding of green proteins are somehow altered with the interaction with surrounding proteins. So this data is quite interesting for me because even such a small, short period of simulation, we could find a sign of destabilization by the protein-protein interactions in cells. Okay, so yeah, that's it for the first point. And second point is more compact conformations. I will just show you just one example, which is a alpha-synuclein in mammalian cells. And the Philip Zelenko group did an incident NMR work on alpha-synuclein. And I think the main theme of this paper is just showing that alpha-synuclein exists in an IDP as if it were in solution state. But they did a further investigation of the protein behaviors by introducing so-called lanthanide binding tags and using the PRE. Actually, PRE has already been shown in the former speaker, and which is very strong method for investigating the behaviors of larger proteins. So I would just again, I would just briefly tell you about how the PRE and other parameters called PCS works. When you introduce paramagnetic lanthanide ions into your protein system, we can use the two very effective and informative information called PRA and PCS. The most important thing is that these informations has the information of long-range distance information. So ordinarily we use NOEs. I will talk about NOEs later, but PRA, PCS very effective for getting information at long-range distance. Okay, so Philip Zelenko's group used the gadolinium ion doped daughter into the alpha-synuclein and found that, okay, they looked at the average radius of gyration, and the conclusion was that alpha-synuclein has approximately 10% smaller radius of gyration in cells. Okay, so I think this is typical, one of the typical effects of molecular crowding to the intrinsically disordered proteins. Okay, and finally, I would talk about conformation changes. But for discussing on this point very in detail, we need to have a method for protein structure determination in living cells by NMR. So in 2009, we luckily determined for the first time on the protein structures in living environment utilizing E. coli as a host cell. And this is the structure TTHA1718, which are from the Thymophilus HB8. And okay, so this is average. This is the structure, but the main criticism against our works is that the high expression level of this protein in E. coli. So it was 3 to 4 millimolar concentration. So we, in order to answer to this criticism and make this method more robust method for the, say, the protein at near physiological concentration, we further developed a method by mainly, I will not talk about in detail of the method development, but mainly introducing some information science related procedures like automated shift assignment by utilizing genomic algorithm or the Bayesian inference based structural refinement or something like that. But anyway, by having the efficient method for the structure determination, we started tackling for the protein structure determination in eukaryotic cells. And for that purpose, we actually selected cultured, we selected the protein expression systems and particularly insect cells SF9 with the vaccial virus, and taking conventional NOE based approach. And for the people who are not familiar about the NMR, I'm just going to say that NOE, a nuclear overhouse effect, are the main parameter which we usually use for the structure determination by solution NMR. So the intensity of NOE is proportional to the R to the -6 power. R is a internuclear distance. So by utilizing NOE, we usually collect the relatively short distance information, proton distance information within the 5 or 6 angstroms. Okay. So I will make all of the method-based topics skipped, and this is the final resulting structure, which was luckily we could determine this structure relatively good resolution. And because this structure was actually calculated with accumulation of various side chain NOEs, so the position of side chain heavy atoms are also well defined, I would say. Okay, so now we have relatively well precisely determined the structure in cells. So this is a good timing to compare what kind of effect happened to the structure of proteins as an effect of molecular crowding inside living cells. So when we looked at chemical shift differences and the difference in NMR signals, we found this region, which is actually color-coded along here, as a relatively bigger chemical shift changes, and the position as also simultaneously showed are slightly bigger RMSD was a position of backbone atoms or side chain atoms. So we looked at we found that the significant NOE information for these regions are actually simply disappeared, and the resulting structure is something like this. So the in SF9 cells are actually presented in blue, and the in reference in diluted solution structure was in red. So we can say that the axis of this alpha helix was somehow tilted against the protein surface. And we concluded that this is because of the hydrophobic, non-specific hydrophobic interaction with this hydrophobic patch of GB1. Oh, sorry, I didn't mention, this is a protein GB1 domain. They're one of the model protein of for NMR methodology development. Sorry, this is GB1. So we concluded that the change in structure may be responsible by the effect of non-specific hydrophobic interaction of that part. But actually, changes in the orientation of this helix has already been predicted by the molecular dynamics simulation by Yuji Sugita. And this is the data, and here the simulation and the structure, the result simulation under the most crowded environments are represented in red, and the native structure was presented in green. So you can find that the alpha helix axis was somehow tilted. So so they described that the destabilized GB1 conformation has a slightly opened core with a hydrophobic pocket between residues T22 on the helix and a T2 on the beach sheet, becoming more exposed to solvent. So two independent approaches, one is in-cell NMR, the other one is molecular dynamics simulations, showed relatively similar result. So we are thinking that so the structure we could observe are actually corresponding to the real change happened in SF9 to the GB1. Okay, so I think my time is almost going to up. So finally, I would say that the 3D structures proteins of less than 10 kilodalton size, pretty small, unfortunately, can be determined in living E. coli, SF9, and genopath oocyte cells. And similar procedure can be utilized for proteins in human culture cells, I think. And for medium-sized proteins, we confirmed that we could get NOE information, but the most important thing is the peak separation issue. So we're better to combine this kind of approaches with the selective labeling in order to get the unambiguous distance restraint for the determining protein structures. For precise structure determination, we still need NOE-derived short-range distance information. But as was shown in the chaperone case in the previous talk, that the PRA, PCS, paramagnetic information must be very, very effective for detecting larger conformation changes, such as relative domain orientations, protein-protein interactions. And what we observed for GB1 in SF9 cells, intracellular crowding environment had apparent effects even on the 3D structure of GB1, a well-structured single-domain protein. So I expect more significant effects may be found on intrinsically disordered regions, IDPs, and multi-domain proteins connected with the flexible linkers. So in future, by accumulating the intracellular 3D structures of proteins, it may be possible to accurately predict the influence of the intracellular crowding environment on protein 3D structures. Thank you very much. I would thank all of my collaborators, particularly as the Teppei Ikera, who did in our group did quite a significant work on the protein structure determination in cells in collaboration with Peter Günther. He's a specialist of protein structure determination. Okay, thank you very much indeed.
Great talk, Yutaka. Thank you. We have a question from Lila. Do you want to jump in, Lila?
Sure, and my question perhaps will not surprise you, but these proteins in the cell are surrounded by chaperones. Chaperones are present in a very high concentration. It's interesting that some of what you see could in fact be a consequence of interactions with chaperones. Have you thought about that, or have you begun to explore it?
Okay, I remember the one publication from the Sebastian Hira and Roland Rick in Nature. They actually investigated the effect of chaperones and the intrinsically disordered proteins, maintenance of its function and disorder. So I agree, the chaperone is very important. I'm very sorry I didn't mention about chaperone roles. I just mentioned non-specific interaction. But of course, chaperone interactions will be very, very effective for the behaviors of intramolecular protein structures, I think.
Well, maybe we can get together and I can give you some chaperones and you can add them to your table.
My pleasure. Thank you very much indeed.
Zeng-Yi, I see that you are also hoping to jump in.
Should we wait for the others to raise the questions? I can save my question at the end, probably.
All right, then I'll ask one from the chat because I think we only have time for one more. Could you comment on the usefulness of determining structures by NOEs, which is difficult, as compared to pseudo-contact shifts, which is faster?
Okay, so yes, I would say that currently we're also using the PCS-PRA as a device for determining the protein, particularly for the domain-domain interaction, relative orientation domain. So I think it's basically these two approaches has a how should I say? Yeah, right. The detecting NOE is very, very difficult because we need sensitivity. But if you are very interested in the precise position of side chains, probably we definitely need NOEs, even though this is very difficult. But if you're interested in, for example, a more larger protein structure changes like a difference in the domain orientations or the rearrange of the domains, blah, 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 I think PCS-PRV will be the first choice. So simply, I was interested in the determination of protein fold of a single domain. My first choice was definitely NOE. But I would say that both approaches should be necessary for the future in CNML works. Does it make sense for as a question, as answer to the question?
Yes.
Thank you, Yutaka. So last but not least, our webinar is finishing today with Dr. Zeng-Yi Chang at Peking University in China. And Zeng-Yi was also one of the organizers for today's meeting. So go ahead and finish this out, Zeng-Yi.
All right. Thank you, April. Yeah, those. All right, two exciting talks before me. Really, I have questions I will save them at the end after my talk. So I will, my talk will be on a different aspect. We use unnatural amino acid mediated photo cross-linking to monitor protein behaviors in the living cell. So I have been choosing to focus on a few stories, very briefly, of course. So one is on the ATP synthase. We found two states in the cell. One is called non-inserted, another is inserted state, based on the epsilon subunit of the complex. Another will be more exciting. I think we found a kind of new structure. We call that regrowth delay body. It's assembly of proteins, and as shown by the green colored stuff, they're formed only in non-dividing, non-growing cells, but they are reversible. They will dissolve when the cell regrows. Kind of interesting. And the third part will be a super complex we identified in the living cell that is responsible for translocating latent out-membrane proteins from the cytoplasm to the out-membrane. So all briefly, of course. And actually, as Associated Protein Science, I want to tell you that we did publish a special issue in 2019, focused on proteins in the cells. So if you are interested, you are welcome to get that special issue. Recently, I wrote an essay entitled "Some Random Thoughts on the Life of Protein Molecules in Living Cells" that will be published in BBRC soon. And I raised many questions. Some covered by the two speakers before, and on imagining proteins in the living cell. And in my opinion, we know very little about proteins in the living cell. We have many, many unknown things, as we heard from the two exciting talks before me. So in our study, we used technology developed by Peter Schul's lab, that is using unnatural amino acid mediated photo cross-linking. For that, we can generate mutations either based on gene carried by plasmid or in the genomic DNA. Sometimes we do genomic DNA mutations. And for that, to introduce unnatural amino acid, as I showed here. So we need two things. One is so-called orthogonal tRNA. Another is called orthogonal amino acid tRNA synthetase. Once we have these two, then the unnatural amino acid will be linked to the tRNA by amino acid tRNA synthetase. And then at the proper anti-codon codon site, we change the codon to TAG stop codon, and the anticodon from the tRNA can recognize that TAG codon, and then the unnatural amino acid will be introduced in the polypeptide, nascent polypeptide. And then afterwards, we can kind of monitor the behavior of the polypeptide since its synthesis from the nascent stage to the last degradation stage, all different stages in the living cell. And you know, the unnatural amino acid, after being introduced in the specific position, they will function normally. Usually, they will not cause any trouble to the protein. So protein-protein interactions will occur normally until you expose the cells by UV light, UV radiation, UV 300 nanometer. Then those free amino acids will be activated and they will form free radicals, and those free radicals will able to form covalent bond linkage with many carbon nitrogen groups, protein or other molecules, form a covalent linkage. So then we can break the cells and characterize and identify the proteins either by mass spectrometry or by immunoblotting. So that method is kind of simple, but it's very powerful, I think, indeed. So we have studied many proteins. I only introduce a few examples. So one is the FTZ protein, and this protein is involved in cell division. In the last stage of bacterial cell division, they form a ring in the middle of the cell at a certain stage, and then the mother cell will become two due to the function of this so-called Z ring, which is assembled by the FTZ ring. It's kind of dynamic assembly and disassembly. So this protein is also found in chloroplast, the mitochondria, which also divide in a similar fashion as bacterial cells. So if the protein is defective, the cell will not divide. They form very long cells. So that's the protein. And the people, the monomer of this protein is a tubulin analog, equilibrium analog, is a GDPase. As you can see, the structure is quite similar. They can form a protofilament in vitro through longitudinal interactions. This has been studied, but they can form all kinds of structures in vitro. And so we try to study how they interact in terms of the lateral aspect. You know, the longitudinal induction has been studied by in vitro X-ray crystallography, but how they assemble further through lateral induction is totally unknown. So we have to do this in the living cell. So by that, we introduce the unnatural amino acid in all different residue sites and performing photo cross-linking to see where they can mediate dimer formation of protein-protein action. By that, we identified multiple lateral interaction services and in living cells. So we even developed a method where the unnatural amino acid will be introduced, called a BPPA or BPA, randomly into the protein in all different positions. We'll first analyze what positions will mediate protein-protein actions before we identify which position it is by DNA sequencing. So this is very kind of method similar to direct evolution developed by Francis Arnold. So back to the FTZ. Of course, people have been studying this protein in living cells, in live cell imaging. But most people focus on studying the protein when the cells are actively dividing. So in our case, we also analyze that in active dividing cells, so the log phase cells, they form dimers, meaning they assembled into certain oligomeric structure. But when the cells stop dividing in the so-called late stationary phase, they no longer interact with each other, so do not form dimer anymore. Instead, they interact with other proteins. So that's kind of interesting. So how they become disassembled, probably by interacting with those other proteins. So, examining the protein in non-dividing cells has never been done. Nobody ever cared about the status of this protein in non-dividing cells, but we did that kind of as a control and found something interesting. So we then try to identify what proteins are interacting with FTZ in the non-dividing cells. And surprisingly, we found the proteins ended in the pellet in those non-dividing cells, although they are all in the supernatant in active dividing cells. So why they become insoluble? That's kind of interesting, And we also analyzed the total wild type cells. We also found this protein without any change, no unnatural amino acid at all. So they're found in the pellet, indeed, in the non-dividing cells. Instead, other proteins like EFTU, which is one of the most abundant protein in bacterial cells, are found in the soluble supernatant fraction, but not in the pellet. Neither the grow year, which is a HSP60, interacting with unfolded proteins, they also ended in the supernatant instead of in the pellet in non-dividing, non-growing cells. So that's kind of interesting why it is there. It's unusual. Then we switched to live cell imaging. Of course, we want to see why they become insoluble in the entity in the pellet fraction. So indeed, in the active dividing cell in the middle, as people always see, a ring in the middle. But in the non-dividing cells, they ended in the two ends, the two poles form two granules, as shown by the two colored stuff very nicely. So, and we can see that by live cell imaging, FTZ linked to green fluorescent protein, or by immunofluorescence microscopy. So then we did more study to see what they are by labeling the different parts of the cell, outer membrane, inner membrane, cytoplasm, and we found such granules formed by FTZ are barely accessible to cytosolic proteins, so impacts space. And if you break the cell, lyse the cells, those structures can maintain. They're still there. They do not disassemble. So, but they have no membrane. So there's no either inner membrane or outer membrane surrounding those structures. So they are membraneless assemblies. So actually, people have identified such aggregations of proteins in non-dividing, late stationary phase cells, indeed, and all assumed to be, misfolded protein forming aggregates. So that's the kind of dead end of those insoluble proteins. But we did quite a few different studies. We found the proteins are folded actually. For example, DNA K and the CLP both bind to misfolded proteins. They are not found in those granules, the green colored granules. But to our surprise, the DNA K forms certain kind of assemblies beside those granules. Probably Lila can show us what does that mean. And but they are different from the granules we saw. And of course, we identified the proteins present in those granules by mass spectrometry. And we found all kinds of proteins, many different kinds, involved in cell division, metabolic enzymes, also subunits of RNA polymerases, amino acid DNA synthetases, and the ribosomal proteins. All kinds of proteins are found, other than the cell division protein FTZ, as shown here. So then we asked whether those, the FTZ proteins are reversible, whether they can come back when the cell regrow, because the cells can be placed in fresh medium, and some cell will grow. And interestingly, we found that the proteins can be dissolved and reused for forming those Z ring. So, and of course, many cells will maintain those granules at the time range we examined. And particularly interesting is that when we let cells divide for some time, for 2 hours, so many generations, we still could identify certain cells maintaining that granules. So shown here. So we consider those cells could be so-called persister or dormant cells, meaning they non-divide, non-grow in the population, although other cells are dividing. So, so meaning the assembly is reversible, the proteins are folded. So that's kind of interesting. And indeed, we found the the formation of these granules will be correlating to both the regrowth lag time. We know bacterial cells always have a lag time when they regrow, either short or long. Can be very long. When they're very long, we call that the dormant cells. So they are sleeping, right? So we have a nice correlation between that and as well as antibiotic tolerance. You grow the cell to a longer time, probably those granules are formed in a more compact structure, theoretically. So they will compact to higher tolerance and longer lag time. So, and they will not be killed by ampicillin. So those are cells keeping the structure, will not be killed. So persistent cells have been identified in 1944 when antibiotics were first applied and used, and certain small percentage of cells are found to be in such a state that they are drug tolerant. They cannot be killed. They're always there, but it's not a drug resistant. It is a non-inheritable state. So when they regrow, the cell become sensitive to antibiotic again. But when you get the next stage of non-growing, they are there again. So that's a very fascinating type of cell. And for this type of cell, the challenging for studying them is so-called persistent cell is that they lack an easily distinguishable biomarker. That means you cannot tell whether it is or is not a persistent, as I showed you in the population. They all look like the same. So you don't know which one is a persistent, which one is not. Most of cells are non-persistent, of course. Only a small portion will be persistent, as I showed that they exist in very low abundance in the cell population. So those are challenges. And until now, people say, the issue is still there, the persistent cells, as elusive as ever. So people don't know how to study that. So once our papers are published in Cell Discovery, faculty opinion recommended that article, and I think that the description is very nice. Said it would be hard to overestimate the importance of this paper. So we're still working on this structure, how they're formed, what is the protein status in the regrowth delay body, and why they are reversible. So those they can be. We are performing both in vivo and in vitro studies to understand more about the nature of those protein assembly in the regrowth delay body. So it's very exciting. Probably I'll give you a talk in 1 year or so. So the ADP synthase, the next talk will be kind of short. ADP synthase is a very famous protein, of course, and but most of the studies are performed in vitro. Indeed, there has been poorly characterized in living cells. So the structure, the model is still here. And it uses a rotational catalytic mechanism, very unusual. And so we focus on one subunit called the epsilon subunit. There are dramatically different alternative structures identified for this subunit. And one is called compact. So the C-terminal will become kind of form a 2 alpha helix compacting together. But in another way, in the complex, these 2 alpha helices will be extended into the alpha beta and the gamma center. So both of these were identified in vitro. We call this non-inserted state comparing to the in vivo state, and this is inserted state. So the problem is whether they exist in living cell, especially this complex structure, and what function they play, what status they represent. So that's critical issue as described by John Walker in 2013, about the coherent explanation for the physiological context for such an inhibitor. This is called assumed to be inhibitory, but the physiological condition is totally unknown whether what the function correlates to. So we used, of course, unnatural amino acid mediated photo cross-linking. So did the mutagenesis, introduced the unnatural amino acid into different positions of the epsilon region, the C-terminal, a stretch of residues individually replaced, and then to analyze, whether the two states inserted, this as shown here, and the non-inserted, as I showed before, are indeed exist in living cells. And surprisingly, when we just culture the cell in an LB medium overnight, we saw both states, both inserted and non-inserted, present in the population. So for that, we can easily identify if the epsilon, cross-link with alpha beta from the top, that means they are inserted. If they do not, the crossing with the C, the bottom subunit, that means they're non-inserted. So by that, we can tell whether the ATP synthase is in the inserted or non-inserted state. So as shown here, many positions, this is a regular gel. So for large-scale preparation analysis of living cells, we developed the whole so-called high-scale, high-throughput SDS page. So on which we can analyze 384 protein samples in one single gel. So by that, we can compare, the changes, whether they are inserted or non-inserted. So then we can see whether residue is in that position or this position. So that is a very powerful technology developed by Yang Liu in my lab. So using that high throughput gel, we analyze all kinds of variants, introducing the unnatural amino acid at different positions on the epsilon subunit, and place the cells under different conditions, all kinds of conditions. And we found that, the non-inserted, the inserted state become hardly, the inserted shown by the alpha beta subunit cross-linking are hardly detectable in the fresh LB medium, as shown here. You don't see there's inserted state anymore, or in the glucose containing M9 minimum medium. That means once you have a good condition, they do not form the inserted state. In a harsh condition, a non-favorable condition, they form the non-inserted state, as shown here. So that's a very powerful technique for us to tell when the inserted state are formed. So, and also we found those two states are reversible, from the non-inserted after being cultured in the medium for a very short period of time, like 30 minutes or 1 hour, they become inserted. They will be extended to the top. So as I showed here, and so by that we can analyze all different conditions to see when they will form the inserted state, when they form the non-inserted state, how they will exchange to each other. So by that, and by to make a long story short, by that we identify two states of the ATP synthase. One is called the non-inserted state. We consider that correlate to a high gear state when the cells are under favorable condition in the fresh medium. We don't detect the inserted state. So then when the conditions become worse, bad, we see a kind of mixed gear. Both inserted and non-inserted will be detected. And when the conditions become harsh, we see the inserted state being dominant. So we call the low gear state. So meaning the ATP synthase activity of ATP hydrolysis, ATPase activity will be inhibited when the cells are under unfavorable condition by having the extended state or inserted state of the epsilon subunit. So these two are equilibrium. Actually, there's a middle state as shown here. They are neither inserted nor non-inserted. So these two levels are not correlating to a total of one. It could be both can be increased. Like they're both inserted and non-inserted can be increased under certain condition. That means there's a third type of state that can switch to the other two. All right. So much for the ADP synthase. The super complex, last one, and that was identified when we tried to analyze the SIR-A protein in living cell. SIR-A is a primary chaperone functioning in the periplasm and for the biogenesis of outer membrane proteins. That's a simple definition. So previous studies based on genetics and in vitro analysis, people built such a model that the nascent polypeptide synthesized by the ribosomes in the cytosol will be translocated across the inner membrane through the Zeng-Y and then they would be taken care by a few chaperones, SIR-A, SKP, or DGP. And DGP, we found that as a proteinase instead of having any chaperone activity. Afterwards, the nascent polypeptide will be transferred to the outer membrane protein complex called the BMA complex, before finally folded and assembled in the outer membrane. So that's the conventional model. We call this stepwise model. So we introduced a natural amino acid at different positions of the SIR-A protein. So SIR-A structure has been determined. It's a very unique structure. It has a P2 domain, not at the end, in the middle, extended to the outside. So this is totally unknown why they have such a structure. So by introducing the unnatural amino acid into different positions, we found, interestingly, that the SIR-A protein will interact with the client nascent protein, OMPF and LAMB, outer membrane proteins, through the end domains, through this blue color domain, all right? But they interact with BMA, the one sitting on the outer membrane, claimed to be important for outer membrane protein assembly and folding through the P2 domain, mainly. So in other words, the P2 domain interact with the BMA, and this domain interact with the client outer membrane protein. So those are based on such in vitro analysis, very powerful indeed. And also interesting, when we introduced a natural amino acid into the nascent outer membrane protein, OMPF or LAMB, we found also very interesting. So those nascent proteins will interact with SIR-A only through their terminal region, either C-terminal or N-terminal region, not the middle region. We did not detect any interaction between the middle region and the SIR-A. So that's also a different, very powerful indeed to identify how they interact in the living cell. So probably this may have something to do for the beta barrel formation, because N and C has to meet each other to seal the barrel, the beta barrel structure of the outer membrane protein. So to make a long story short, we identified, as I mentioned, yeah, there's the tertiary complex, BMA, SIR-A, and outer membrane, nascent outer membrane proteins. And more interesting, we detect a large complex using a blue native gel with about 400 kilodalton. And here we found BMA, SIR-A, Sec-Y, or Sec-E. So in this band, as I showed here, different antibodies will all interact with this this band. So meaning there's a large protein complex here. And so based on those studies, we proposed a super complex model, which will be responsible for the translocating the outer membrane protein from the cytosol across the inner membrane, the periplasm to the outer membrane. There's a one super complex. There's a so instead of stepwise, you know, and this will be a safe channel for the nascent unfolded outer membrane proteins. And recently we identified another interesting novel form of a protein. We call this Sec-AM, because they are represent an N-terminal, about 400 residues N-terminal of the Sec-A protein. So that's a kind of truncated form. And so from our in vivo study, we could not detect the interaction between the outer membrane proteins and the Sec-YG. That's the conventional translocal for the outer membrane protein people assign. Instead, we found outer membrane proteins interact with Sec-AM. So we consider that to be the translocal for the outer membrane protein. And indeed, we detected interaction between the periplasmic protein and the Sec-YG. So probably they are different. They have different function. One is responsible for translocating outer membrane protein, and the other for the periplasmic protein these two types are very different actually in feature, in structural feature. So it's reasonable for them to have a different transportation channels. So, and in this 25 minutes, I showed you the ATP synthase forming two states in the living cell. One is high gear, one is low gear when the cell conditions are different. So totally under natural conditions. That's kind of interesting. Another thing is a regrowth delay body, which will be formed in non-dividing, non-growing cells, but they can redissolve and come back. The proteins can be reutilized, and this kind of reversible, cyclable form of the protein. And last but not end, we identified a supercomplex involving BMA, SEC-A, SEC-YG, and SEC-A, and found a new form of SEC-A, SEC-AM involved in the biogenesis of outer membrane proteins. And we also studied some other interesting proteins like the acid resistant chaperone, HDA. And this protein has a disordered structure when they're functional, so totally the reverse. And it has a chaperone protecting chaperone mechanism. And indeed, those are unnatural amino acid mediated photo cross-linking told us a lot of very interesting stories. One thing that I did not talk about is as Yutaka mentioned, we found proteins in the liver are very dynamic, extremely dynamic, some proteins. So for mediating protein interaction, usually for some proteins, you have to be into the right surface interface of the subunit. But for other proteins, we can choose at any position, they can mediate diamond formation. So we published a PBRC paper. There's a striking result. I think after listening to Yutaka's talk, I think probably proteins in the cells are very dynamic as we assume, totally dynamic. So the meaning is totally unknown. So we have a lot to learn about the protein. And with this, I want to thank the students who did the work, Dr. Jiayu Yu, Dr. Yan Liu, and others, Yan Wang and the Rui Wang on the supercomplex, and Funjin on the SEC-AM, so the regrowth delay by and Jiayu and Yang Liu, also the high throughput SDS-PAGE technology by Yang Liu and Jiayu. And with that, I want to end here, and we'll be happy to answer your questions.
Thanks, Zeng-Yi. I know we're a little past the scheduled end, but we do have some questions. And of course, we can stick around and have any discussion as long as we have it. Lila, do you want to get started?
Sure. I'm fascinated, well, by many of the systems you described, but in particular, the SEC-A. What is the difference? I may have missed this between SEC-A-N and SEC-A.
By mass spectrometry at this stage, we could not purify enough protein for mass spectrometry, identify exactly. But we found the SEC-A-N represent the 375 residues of the total SEC-A protein, which is about 1,000 residues. So it's kind of 1/3 of the total SEC-A.
And you think it's the N-terminal region?
N-terminal, because by antibody analysis, also mass spectrometry, it corresponds to the N-terminal. Yeah.
Thank you.
And we have one from the chat. Thank you for such a wide encompassing work. How do you judge the usefulness of photocross-linking agents based on unnatural amino acids to analyze LLPS phases as compared to more specific high affinity interactions?
That is an interesting question. So to use unnatural amino acid to distinguish the liquid phase separation and the high affinity interaction. Indeed, in our case, for example, FTLZ, when they form the regrowth delay body, it's kind of a separation, it's phase separation, but phase separation is a fuzzy term. People talk about that, but nobody can define what is phase separation. Indeed, is amazing. Many proteins can assemble together. They can form interactions, as I showed in one PPT. The FTLZ will interact with other proteins instead of itself. And also, by such cross-linking, we can tell that proteins are folded. Usually people do not know whether they're folded or not. So we introduced a natural amino acid into different spatial positions to see the pattern, whether they are similar when they are in the same location and different in different locations. If they are random interaction, then they should form dimers always without exception. So, but the high affinity, low affinity, that I know, but the specific, non-specific interaction we can detect with unnatural amino acid photo cross-linking to a certain degree. But that's a kind of qualitative analysis. It's not a quantitative, how tight they are. And although we say we can capture transit interactions, but that depends a lot of issues. We do usually we see a very faint band, because those represent very transient intercellular interaction. So some reviewers cannot understand why it's so faint. But in our case, we have to compare the control and the positive UV exposed cell. If there's a band, means there is inaction, could be very transient. So yeah, quantitatively we can use them, but quantitatively no. We have to do in vitro studies. Yeah, I'm not sure whether that's correct. Yeah.
And we have one more from the chat. Are these granules formed by phase separation events? And this question popped up relatively early in your talk.
We consider there's this a type of phase separation, but it is on the semi-liquid. It's kind of a solid state. They are very stable. We can break the cell, they are maintained, and we try to examine their structure using electron microscopy and others. It's not an easy thing, They do not form a regular structure. They have different sizes and different shapes. So we don't know how they are assembled, although they are folded in our term. They're folded, they have a folded structure, but how they interact with each other is totally unknown. So at this point, but yes, it's one type of phase separation, but phase separation is a fuzzy term. It's a fuzzy term. We don't know what is phase separation, but proteins have such properties to interact in a kind of non-specific fashion. I think in my BBRC article, I discussed about this issue extensively. So I think it's very important for protein evolution. And a certain stage, protein can form such a phase separation structure and allow protein to form folded structure, functional folded structure to be evolved. Yeah.
All right. And are there any lingering questions that our speakers want to ask each other?
Yes, sorry, I have some questions. For both Lila and Yutaka, I think, yeah. When we think of protein folding, there are two aspects. One is thermodynamics, right? Free energy difference between the folded and unfolded state, as you showed the funnel. Another is dynamics. Proteins are moving all the time. So in nanostock, I think in the HSP70, they have a promiscuity to bind proteins in a different way or different affinity. So I'm wondering whether that's a kind of balance between thermodynamics and the dynamics. That's how proteins are in the living cell or in vivo, how protein function. Proteins can perform so many different versatile functions. They have to have a certain, like HSP70, they can bind to so many different forms of protein. So what is how do you think about the balance between these two? And I think there are people arguing which is more important, thermodynamics or dynamics or for protein functioning. Some people say dynamics is more important than thermodynamics. So in my mind, it's always the free energy difference is so small between the folded and unfolded. So that means dynamics is also important here in protein, structure and the function. How do you think? I'm not sure whether I described the question clearly. Lila.
I'll take a whack at that, but briefly, which is that I think living systems are not at equilibrium. They are definitely consuming energy and their flow systems. We did a model, it's in a Cell Reports paper, I can't remember which year actually, but with Evan Powers, of the chaperone networks in E. coli and the folding process. You take a folded protein and you put it into that network and see what happens. But we did it with a couple differential equations because this is a kinetic dynamic system. It's a flow system. So I think you're right. You shouldn't really get too fixated on thermodynamics, although it's instructive to know what the landscape looks like. But the other comment I would make is that everything in the cell is spatially organized. And so you have to be cautious to not extend from what we learn in a homogeneous medium and apply it to the cell. And for the HSP70 system, the J proteins tend to be localized, and the 70's job is governed by which J it interacts with, which puts it in a location which says it will see those clients. So it's not like a big bag by any stretch. So those are my comments, but I don't know. Yutaka may have something else to say.
Well, it's very, very difficult problem. And I'm very sorry, I have not particular answer to your questions.
Yeah, so you talk about our BBRC paper. We analyzed two proteins. One is a small histoprotein called IBPB, and another is the DGP that I showed as a proteinase. You know, in living cells, we were surprised when we tried to identify the interaction surface. So we introduced the residue at all positions, all individuals across the whole polypeptide chain, and all positions can, mediate interaction. They form dimers. That's totally unexpected. You know, based on structure, only certain residues can form dimers. But we identify it's not a misfolded protein. They are folded protein, but they can do this. So that by that we draw the model showing the proteins are very dynamic. Like a human person, you can change dramatically to mediate interaction. So as usual, indeed in the cell they're very dynamic. So why it seems to me they're more dynamic in the living cell than in the solution? That's surprising. Indeed, we could not explain it, but the fact is there. The result is there. So why they're more dynamic in the cell?
Well, my impression is that I'm not so sure whether the protein behaviors in cells are more dynamic, but we can say that the protein behaviors in living environment is more heterogeneous, I would say, because the interactions are so heterogeneous. We have like a chaperone interactions or the other non-specific interactions or specific interactions. So I think it's we can see the image of the average structure, but anyway, in cells, I think its protein behaviors are so heterogeneous. This is my impression.
Okay.
So Yutaka, I have a question for you on your GB1 structure determination in E. coli. So you may have said it, but what was the actual concentration in E. coli, and what was the concentration in your NMR sample as a whole?
Okay, so in our the initial result on in E. coli, the concentration, as I mentioned, is very high, 3 to 4 millimolar concentration in NMR tube. And the last example I showed as a GB1 in SF9 cells, we estimated the intracellular concentration might be 50 micromolar, something like that.
I see. So in your NMR tube, it would have been much more dilute than that. Right?
I think so, yes.
That's amazing.
So we definitely need special equipment called bioreactor to continuously providing the fresh medium into the cells, and that's why we could get a quite long duration NMR experiment. That was a key for the determining the structures based on the such insensitive NOE based informations.
Thank you.
All right, so I think that we are coming up on 2 hours, so I think we have to close it out. Chuck, I think you wanted to say a couple things to end us today.
I have one last really quick question for Lila. Okay. So Lila, how many HSP70s do humans have? How many different ones?
So we actually have 12 or 13. It depends who you ask. And as you probably know, there are 2 major HSP70s in the cytoplasm, one of which is constitutive, so it's present all the time, HSC 70, and the other HSP A1 is stress induced. We're looking at both of those, how they bind. They're very homologous. I think they're 86% identical, but not absolutely identical. And we see some differences in their allosteric behavior. That's in a PNAS paper from a couple of years ago. And then the other HSP70s are in smaller amount or in organelles in eukaryotes. So they have more specialized functions. They're all their nucleotide binding domains are quite homologous, and substrate binding domains not so far apart either. So it's the mechanism is conserved in all likelihood, but there may be subtle differences in substrate binding.
And are there lots more
Many more J's. Many more J's. Very good question. Sorry, I should have said that. The J's are the diversity factors.
Yeah.
They allow specialization, and they're really quite variable. There are 3 classes, and they have different abilities to bind substrate. They have domains that differ, domain architectures, a lot of different possibilities for functional specialization come from the J's.
Okay. All right, well, I think we will wrap up here, but I just want to thank everyone who contributed to the talks today and to the questions and answers, and who helped to organize this event. These were 3 absolutely spectacular talks, so we're really grateful to our speakers for coming. And let me just say in closing, if anyone is interested in organizing or even suggesting the topic for a webinar, we're always looking for suggestions. So just get in touch with me or the society, and we'd be happy to work with you on this. And so I think we'll stop here, but I just wish everyone a really fine evening or morning, and we'll see you hopefully at the next webinar. So bye-bye, everyone.
Bye-bye.
Thanks,
Bye-bye.
everyone.